Hi, welcome to AP Biology. I'm Allison Kate, and today's lesson is going to be on the cell cycle and the regulation of the cell cycle. We're also going to talk about Lab Investigation 7, just the first part about mitosis, and the cell cycle. We're going to look at two science practices today. We're going to perform a chi-square analysis to test a null hypothesis. And we're also going to look at predicting the causes or effects of a change or disruption to one or more components of a biological system based on biological concepts or processes. And as you might guess, these are going to relate to the cell cycle and its regulation. By the end of this lesson, you'll be able to describe the events that occur in the cell cycle, explain how mitosis results in the transmission of chromosomes from one generation to the next, describe the role of checkpoints in regulating the cell cycle, and describe the effects of disruptions to the cell cycle on the cell or on the organism. All right, so let's start with our warm-up, which is from yesterday's lesson on cell communication. Yesterday was kind of the nuts and bolts and overview of cell communication, and today we're going to take a look at some pathways that actually regulate the cell cycle. So let's take a look at the question. You're looking at a model or a diagram that represents a generalized hormone signaling pathway. You are asked to briefly explain the role of each numbered step in regulating target gene expression. So you can see you've got step one, and step one is labeled on the plasma membrane and you're given direction. You can see that on one side you've got the extracellular fluid, and then on the other side you've got the cytoplasm. And so step two takes place in the cytoplasm and it has multiple steps. And then finally, we see step three is occurring inside the nucleus, and this must be a eukaryotic cell because we see a nuclear membrane with nuclear pores. And step three does something with the DNA. All right, so if you did your homework last night, good for you. Take it out so you can compare your answer with the scoring guidelines. And if you're just joining us now, think about how you would answer this question. Okay, so 3 steps. What are they and what is their role in regulating this response? Okay, so first of all, step one. Step one is called reception, and reception happens when a ligand, in this case it's a hormone, binds to a receptor that is on the surface of the plasma membrane. Once that ligand or hormone binds to the receptor, we see a shape change in that receptor molecule. Okay, so that kind of marks the end of reception with a shape change once we've got the signal molecule bound to the receptor. The second step, again, occurring in the cytoplasm is called transduction. And this is a multi-step pathway that amplifies the signal. So you can have one signal molecule that is going to generate a lot of other molecules that are going to bring about a coordinated response. Step three is called the response. And in the response, in this case, we are going to target the expression of a gene. It could either be stimulating gene expression or it could be stopping or repressing gene expression. So those are the 3 steps: reception, transduction, response, and they're characteristic of any cell signaling pathway. And as I said, today we're going to apply this to cell cycle regulation. All right, so let's get started. Here's what you need to know. In eukaryotes, there are really 2 processes that we see in the transmission of genetic information, and you've probably heard of both of them before: mitosis and meiosis. In eukaryotic cell division consists of mitosis, which is the division of the genetic material in the nucleus, followed by cytokinesis, which is the division of the cytoplasm. Okay, so that's mitosis. Gametes are produced by a variation of cell division called meiosis. Meiosis yields non-identical daughter cells that have only one set of chromosomes, which is half as many as the parent cell. And tune in tomorrow because tomorrow we're going to focus on meiosis, whereas today we're focusing on mitosis and the rest of the cell cycle. All right, so let's take a look at this diagram. The cell cycle is a highly regulated series of events that is used for the growth and reproduction of cells. It consists of sequential stages of interphase, which is in turn made up of 3 parts called G1, S, and G2, followed by mitosis, which is the division of the nucleus, and cytokinesis, which is the division of the cytoplasm. And as you look at this diagram, you can see that the majority of the time is spent in interphase, and that only a small portion of the cell cycle is the actual nuclear and cytoplasmic division. Okay, so briefly, the G1 is the general growth phase of the cell. The S phase stands for synthesis, and this is where the chromosomes are replicated or duplicated. And then G2 is the last part of interphase, and this is where the growth in the cell is specifically about manufacturing molecules and creating energy molecules to use through the process of cell division. And then finally, we have the M phase or mitosis, where the chromosomes are distributed to the new cells, and then the cytokinesis divides, creating two new identical daughter cells. And we're going to talk about how that process happens next. Okay, so let's just take a look at how the chromosomes are actually passed on to the daughter cells. And I tell my students when I'm teaching in the classroom that if you don't remember anything else about mitosis, the outcome of mitosis is to create two genetically identical cells. And you're going to see why that's important in some of my examples, but we want to create two genetically identical daughter cells or cells. Okay, so first of all, just to look at this diagram, we're only looking at one chromosome, although we know that particularly in a human cell, we'd have 46 chromosomes. We're just looking at one to simplify it, and it's represented here in purple. It has not yet duplicated. Okay, you can also see that there is an area that is a constricted part, and that's called the centromere. And then you can see we call these the arms of the chromosome. So then the chromosome is going to duplicate, and that occurs in the S phase of interphase of the cell cycle. Once duplicated, a chromosome consists of two chromatids. They are connected along their entire length, and each chromatid contains a copy of the DNA molecule. Finally, you can see that through molecular and mechanical processes that we'll go over in a minute, the sister chromatids are separated into two chromosomes and distributed into two daughter cells. And these are genetically identical. The DNA copy is identical, and so those two new cells are exact copies. So mitosis plays a role in growth of a cell or an organism, if it's a single-celled organism, tissue repair, and asexual reproduction in single-celled organisms. So let's take a look at either cell reproduction, that's the first image we see, or if it's a new organism that reproduces asexually, I have an image of binary fission, which is used in bacteria. This example is an E. coli, and the end result after the genetic material is copied and the cell splits, we have two identical E. coli bacterial cells. In B, you can see that we also use mitosis in growth and development. So just after an embryo is formed, an egg cell fertilizes, a sperm cell fertilizes an egg, you get a zygote, and then it starts to develop into an embryo. All of those divisions are mitotic divisions. So we want identical cells up until a point where the cells begin to specialize. So you might see this in an animal cell as a blastula stage. And then finally, mitosis happens to renew tissue that is damaged or worn out. And so, for example, one of the reasons you want genetically identical cells here is, let's say you scrape your knee and your tissue needs to get repaired and replaced. You want the tissue that is replacing those damaged cells to be exactly the same. So again, we use mitosis for just cell growth, organism growth if you're single-celled, tissue repair, and asexual reproduction. So now we're going to talk about mitosis, which is the actual nuclear division. And we don't really need to know all the details, but I want you to be able to recognize all of the stages. In my class, I do something called a handy model, and I'm not sure if you can see it here. I don't know if you can see my hands, but you can follow along with me. So interphase, make a fist, right? Interphase, the cell is not dividing. And then in prophase, the chromosomes condense and they shorten and thicken, so wiggle your chromosomes. In metaphase, the chromosomes are going to line up in the middle of the cell. In anaphase, they're going to pull to opposite sides. And then in telophase, we get our cytoplasm division, and you have two genetically identical cells. So that's the handy model. But let's just kind of take a look at some of the important things. So G2 is the last part of interphase, and it's characterized by the fact that you can still see an intact nuclear envelope and one or more nucleoli. You can also see the chromosomes are unwound. They are duplicated, but they are not condensed. So they're kind of fine threads that we refer to as chromatin. And I'm not going to read this to you, but it will be here if you go back and look at these slide decks later on. In prophase, what you start to see is the nuclear envelope begins to disappear and the chromosomes begin to shorten and thicken and condense. You can see that they're held together at that centromere. And then you can also see the formation of the mitotic spindle, which is made of microtubules, and it's super important in actually moving the chromosomes during the process of cell division or nuclear division. You can also see these little asters, which are radiating out from centrioles. And they seem to play a role in kind of the physics or mechanics of bracing those microtubules or spindle during this process of moving chromosomes. One note, centrosomes are only found in animal cells. Plant cells do have centrioles, but they don't arise from centrosomes. Then we get metaphase. Prometaphase comes just before metaphase, and this is where you can really see the spindle fibers beginning to form. And there are two kinds of microtubules. There are microtubules that attach to the kinetochores, which are the little protein discs that are at the center of those sister chromatids. So you can see that they are actually attaching to the kinetochores, and then you have non-kinetochore microtubules which do not connect to those chromosomes. Okay, and then what you can see in metaphase is meta means middle, so those chromosomes are maneuvered to line up right in the middle of the cell at the equatorial plate, and you can see the spindle is composed of aster fibers, non-kinetochore fibers, and kinetochore fibers, and the fibers are actually microtubules. Okay, so then we see that the anaphase is next, and anaphase, you see the chromosomes, chromatids are being pulled to opposite sides of the cell by those kinetochore spindle fibers. And what's happening to move them is there's actually motor proteins that actually will pull toward opposite sides of the cell. And then finally, we get telophase and cytokinesis. And you can see that the non-kinetochore microtubules are important in this stage at pushing them apart to opposite sides. You can also see a cleavage furrow beginning that is going to actually separate those into two new cells, identical sister cells. And you can see that in telophase, you kind of get a reverse of prophase where you start to see the nucleolus is reforming, the chromosomes are unwinding into the chromatin form, and you start to see that nuclear envelope reforming. And now you can also see that the microtubules in the spindle apparatus has been dismantled. Okay, so that's mitosis, that's the cell cycle. Before I talk about regulation, I want to talk about a special stage of the cell cycle that is called G0. And this is kind of a suspended animation stage where it no longer divides. So it can exit the cycle into the G0, but it can re-enter the cell cycle in response to appropriate cues. And I'm going to give you some examples of what that might be. So non-dividing cells may exit the cell cycle or be held at a certain stage in the cell cycle rather than continuing through to divide and create two new cells. So what we're looking at, and I'm going to go into detail in the next slide about checkpoints, but you can think of them as stop and go signals. So what you see here is a G1 checkpoint that has a red light. And what that means is it's not getting the appropriate cell signal to pass through to the next portion of the cell cycle into the synthesis phase in this case. And so it will actually exit the cycle and go into this non-dividing state called G0. So let me give you a couple of examples. Mature neurons or nerve cells are an example of a cell that is in G0. So that means that once a nerve cell matures, it will not divide. It will no longer go through its cell cycle. In contrast, epithelial cells like your skin cells or the cells that line your digestive tract, they're constantly dividing. They're replacing the layers of cells. So the outer layer of your skin is actually dead epithelial cells, and below that you have the cells that are dividing to replace those. And then finally, we have liver cells, which are an example of a cell that enters the G0 stage, but it can be induced to re-enter the cycle and divide. And that's usually done through a physical means. So for example, if you wanted to donate a portion of your liver for a liver transplant, they will actually surgically remove a lobe of your liver, and then that incision that is done surgically will stimulate your liver to regrow. Okay, so those are three examples, ones that don't divide, ones that constantly divide, and the ones that don't divide unless they have a special cue to cause them to re-enter the cell cycle. All right, so let's talk about checkpoints. Okay, so you can see in my first image that there are 3 checkpoints. They're kind of these red blockades that you see popping up. So they are called the G1, the G2, and the M checkpoint. So in the G1, for many cells, the G1 checkpoint seems to be the most important. Sometimes it's referred to as the restriction point. And what's checked at this point is the size of the cell. Is it adequate? Does it have growth factors? And the environment. And the environment is important in that not only do we have internal controls, there are external controls of the cell cycle as well. And my last slide will talk about external or environmental factors that can affect the cell cycle regulation. Okay, so the G1, again, is the first checkpoint, and it turns out that if cells pass that G1 checkpoint, they do tend to go on, they synthesize their DNA, they prepare to divide, and they divide successfully. But it's also the place that cells can drop out into that G0. Then the G2 checkpoint, and this gets a little complicated, so kind of hang in there with me because there's a lot of acronyms. So in the G2 checkpoint, there is a chemical that is called MPF, which stands for mitosis promoting factor. And it's actually a combination of 2 other molecules, an enzyme called cyclin-dependent kinase, or CDK for short. And you might recall from yesterday's lesson that a kinase is an enzyme that phosphorylates a molecule. So it adds a phosphate group to a molecule. So anytime you see a kinase, that's the job that enzyme is doing. And another protein called a cyclin. And in a later slide, I'm going to show you the relationship in terms of what's going on in the concentrations of CDKs, the cyclin-dependent kinases, and the cyclin proteins, because they are related. When they join together, they create this mitosis promoting factor, or MPF for short. And it controls the movement of G2 into mitosis. And the way it does that is by phosphorylating and activating proteins that are involved with 4 different aspects. Chromosome condensation, which is necessary for mitosis, right? You need your chromosomes. Nuclear envelope breakdown, which happens in prophase, because you need to be able to move your chromosomes without that nuclear envelope being in the way. Spindle assembly, how do you move the chromosomes? You need to have your microtubules. And then it actually creates an enzyme that will break down that mitosis promoting factor itself to signal the end of that mitotic phase. So this one's a little complicated, but it is the checkpoint that is between the second growth phase and the actual division of the nuclear material. And then finally, there's a checkpoint called the M checkpoint that happens in mitosis. It happens between metaphase and anaphase. So I want you to think about what happens between metaphase and anaphase, right? In metaphase, the chromosomes line up and the kinetochore spindle fibers attach to them. Okay, so if those fibers do not attach to the chromosomes, anaphase won't begin because there's no way to move those chromosomes. So the M checkpoint is to make sure everybody's attached. All right, a little bit more about that M checkpoint that I think this picture really illustrates it nicely. On the left side, you've got a red flag. You are not going past that. And that's because if you look carefully at the image down here, those spindle fibers are not attached to all of those chromosomes. If you look at the green light, the signal to go, you can see at metaphase, if you look carefully, all of the kinetochore fibers are attached to those chromosomes. And so during anaphase, they can be pulled to opposite sides of the cell. This ensures that you get equal distribution of chromosomes with all of the genetic complement in each of the new cells. So let's take a look at the interaction between cyclins and CDKs. Let's look first here, because this is a really good image to start with. The purple line is the MPF activity, and notice that it's low and it peaks at mitosis and then it drops off again. And then it's low and it peaks at mitosis and it drops off again. And then it peaks at mitosis and drops off again. So you notice that MPF is active during mitosis, okay? Now, if you look at the cyclin line, cyclin cycles, right? So we can see that it builds up prior to mitosis, and then it at its peak, and then it declines rapidly. Then it builds up in the next cycle until it reaches a peak in mitosis, and then declines right after. Same thing again. So now let's look at it in this cycle form, okay? Just to orient you, here's your G1, your S, your G2, and your M. Remember that MPF is working at that G2 checkpoint, which is going to give you the signal to go ahead on into mitosis and divide. So cyclin, gets its name because it cycles, right? So here it is fully built up. But then, and let me come back to how it degrades over here. But so you get your cyclin building up. CDK is around the whole time, but when cyclin builds up to a critical level, they combine together and form mitosis promoting factor, which is the signal that says, okay, go ahead on into mitosis and divide. Then what happens is that the cyclin is degraded and CDK continues to be around, and it will pick up the cyclin in the next cycle. And so that is how we pass from the G2 to the M. So remember that those are all the internal signals that happen to regulate the cell cycle. There are also external factors. And I'm going to give you an example of a growth factor. This growth factor is called platelet-derived growth factor. Platelet-derived growth factor, right? PDGF. That's another acronym. And it will stimulate these fibroblasts, which are cells, to divide in culture. So you can see those cultured fibroblasts. If you add that growth factor, then they will divide. Okay. There are two other things that I want to show you, and these are external signals. One of them is called a density-dependent inhibition, which happens when cells that are really crowded, there's not enough resources for all of the cells, they usually will stop dividing, so that it gives the cells that are already there the best chance to survive. So when cells get super crowded, they stop dividing. Also true of most animal cells is that they need a substrate to grow on and able to divide. And this is called anchorage dependence. So the cells are actually anchored to a substrate. In the Petri dish, you can see they're not crowded, they're anchored to a substrate, and they're dividing. In this case, this is a case of a cancer cell. And what's happening is these cells don't care that it's crowded, and they don't care that they're not actually dividing on a substrate. So cancer cells will exhibit neither density-dependent inhibition nor anchorage dependence. So those are external factors, growth factors, and density, the density that the cells find themselves in. Okay, so there are two types of disruptions I want to talk about. Disruptions to the cell cycle. So if you miss an internal signal or an external signal, it can result in cancer or in something called apoptosis, which is programmed cell death. And this is not necessarily a bad thing. I'm going to give you an example that has happened when your fingers and toes were forming. And so apoptosis is a type of programmed cell death in which the cell components are disposed of in an orderly fashion without damage to neighboring cells. So when your digits are forming, your fingers and toes, you can see that they form with a whole wedge of tissue. And then apoptosis happens specifically in the interdigit tissue so that you get nice fine digits. If anybody has webbed fingers or webbed toes, you know that that apoptotic pathway kind of stops short. Okay, the second thing I want to talk about are disruptions to the cell cycle that may result in cancer. And I want to talk specifically about a transcription factor that's called P53. So first, I'd like to talk about this pathway with that transcription factor present. And then in the image below, I want to talk about what happens without that P53 transcription factor. So we all know that our DNA can be damaged by the environment, especially if you're out sunbathing too much. You know that there is a chance that your cells can be damaged in your skin and you can develop skin cancer. So we've got UV light coming in, causes damage to the DNA, and we see a transduction pathway. So that's the signal. We see a transduction pathway that includes some protein kinases that are going to eventually activate P53. P53 then goes to the nucleus and creates a protein that inhibits the cell cycle. Okay, so we got our signal, we've got our transduction, and we have a response, which is to produce a protein that inhibits the cell cycle. So that looks like the response is that the cell cycle is inhibited. So the damaged DNA is not replicated, so it stops in that cell. The P53 is stimulating the expression of that protein, which is a CDK inhibitor. So it's going to actually inhibit those CDKs or cyclin-dependent kinases, and it prevents the progression of the cell cycle, which means no cell division. All right, so now let's look at what happens without that P53 transcription factor regulating the cell cycle. We see the same signal. Light is damaging the DNA. We see the same transduction. We get our protein kinase cascade going. However, P53 is not activated. So there's some type of a missing or defective transcription factor, which means that we will not be able to make an inhibitory protein, and the cell cycle is not inhibited. So that means that the mutation that happens prevents the activation of P53 so that CDK cannot be, the inhibitor cannot be stimulated, and the cell cycle progresses with the damaged DNA. So that damaged DNA will then be passed along to the next cell in that cell lineage, and you see increased cell division. Okay, so now that you're armed with information about the cell cycle and how it's regulated, we can investigate this in the laboratory. And hopefully some of you have done this in the classroom. Different instructors do different types of versions of this lab. I'm going to share with you the one that I do with my students. The objective of this lab is to root onions in different environments and then determine the number of cells that are dividing. So you're going to look and see how many of them are undergoing mitosis, and then compare them to cells that remain in interphase, that remain undivided. In this lab, the onions are rooted in 3 different environments. Caffeine, which is thought to slow the cell cycle. And if you think about the old wives' tale about caffeine stunt your growth, that you can kind of remember that. And lectin, which is a natural growth accelerator that's found associated with the roots of some plants. So it should increase the cell cycle, and then distilled water is used as a control. So the image there is showing you some different, what it looks like when you root those onions in different cell environments. And so we start this with a null hypothesis and an alternative hypothesis, because ultimately we are going to test to see if our data is statistically significant using a chi-square analysis or goodness of fit test. So our null hypothesis is that there's no difference in the number of cells in mitosis in any treatment. In other words, caffeine, lectin, and distilled water affect the cell cycle in the same way. An alternative, and this is just one alternative, you could have several alternative hypotheses here, but we're going to say that the treatment in lectin will have more cells in mitosis than the control. So we're going to compare lectin to the control, and we're saying that we think there should be more cells that are dividing, which is measured by mitosis, than the control. I want to just share with you that we really are just going to look at the tip of these onions, and that's because if you look at this diagram, we're looking in this rapidly growing region of this root. This is the root cap, so there's not going to be any cells dividing here. And then up here, these cells are already mature, and so they don't divide anymore. So we're looking here in what is called this zone of elongation and zone of cell division, okay? Really in this area. And so what's going to happen is students will root these onions, and then we will take a tip of the root and do a squash preparation of the onion root tip and look at it under the microscope and try to identify as many cells as we can that are dividing and not dividing. Okay, got that? All right, so this is what it looks like. And of course, there's lots of practice before this with identifying these cells. And really, I don't care if the students are able to identify the different stages of mitosis, only that the cells are dividing. But if you can identify the different stages, then that's awesome. But so first of all, cells that are not dividing are an interphase, and they look like this. And the telltale sign is that they've got, nucleoli. This is probably a better example of that, and an intact cell membrane. Okay, and you can't really see the chromosomes, right? They're still in that diffuse chromatin form. And then mitosis. And in this one, we see a cell in anaphase, and you can clearly see those chromosomes are moving apart from one another. There's a bunch of other examples. You can see a cell here, and you can see a cell here that are dividing. Here's one, this is in prophase. So you can see. So basically what happens is students make their slides and then they count up the number of cells they see dividing and not dividing. Okay, so this is some sample data, and this is actually the treatment group. This should say lectin versus the control group. Okay, so I'm going to talk about what we're going to do with this data in just a sec. First, I want to introduce you to the formula that is used to calculate chi-square. And this is straight from the AP Biology formulas and equation sheet. X is the Greek letter X is the chi is the Greek letter X. And then we've got sigma, which is also the Greek letter for S. So got some Greek symbols here. So chi-square is equal to the sum of, which is what sigma stands for, the observed data minus our expected data squared over the expected data. And it is very important that you follow the order of operations here, okay? When calculating a chi-square value, you need to be aware of something called a critical values table. It has two components to it. The first one is called the degrees of freedom. And you can see there we've got 1, 2, 3, 4, 5 degrees of freedom in this chart. They go all the way up to, 50. But we're only going to be working with 2 categories. And the degrees of freedom is the number of categories minus 1. And I'm sure that the stat people will have a much better explanation for this. But it restricts the number of choices that you have, the number of categories that you have. Let me give you an example. So when I teach students chi-square, I do an activity with M&Ms. And I have all the different colors. I believe there's 6 colors. And I'll go to the first student, I'll say, choose an M&M, and they may choose a green one. Go to the next student, choose another one, they choose a blue one, and so forth. And if I come down to the last student, there's only one color left, and they don't have a choice. So that's the -1 part of it. Okay, so the degree of freedom is we have 2 categories, interphase and mitosis. -1 gives us a degree of freedom of 1. Now, the probability that we're going to use is 0.05. And we use this because our kind of bar of goodness of fit is 95%. So we're going to be 95% certain of the whether or not we can accept our null hypothesis. So over here is a table for working out your chi-square. What we're going to do is we get our observed data straight from our data table, and then we have to calculate our expected cells. We don't really know how many to expect, so we have to base that on our control group data, and I'm going to show you how to do that in a minute. Then we go ahead and calculate our chi-square, and we compare it to our critical value. And that will allow us to either reject or fail to reject our null hypothesis. And the reason that we do this is, or how we decide this, is that the chi-square value, if it is greater than the critical value, we have to reject our null hypothesis. There is a difference. If we fail to reject our null, it's because our chi-square value is less than our critical value, and then we are going to basically fail to reject as accept our null. There is no difference in our observed and expected data. All right, so let's go through that. So first of all, students have counted in their control group, which is rooted in distilled water, 186 cells in interphase and 14 in mitosis for a total of 200 cells. And in the lectin treatment group, they have counted 146 cells in interphase and 54 in mitosis out of those 200 cells. So how do we figure out our expected results? So to do that, you're going to calculate the percentage of cells in interphase and in mitosis. So first of all, how do you do that? You would take 186 divided by 200, and then that percentage or frequency is then multiplied by the percentages of the total number of cells in the treated group. So when we look at that, we see that we've got 186 cells out of 200, which gives us a frequency of 0.93, and we multiply that by the total number of cells in the lectin group in interphase. And we can see that we get an expected value of 136. Let's do that again for the mitotic cells, right? So we had in the control 14 out of 200, which gives us 0.07, and then we need to multiply that by the total number of cells in the treated group, which is 54, and then we get 26. So then we have to do our O minus E squared. So we see 186 minus 136 squared divided by 136, and that gives us a value of 18.3. We repeat that for the mitotic cells, and we get a value of 30.1. So remember that the chi-square value is the sum of both of those, or all of those. You could have more categories, but in this case, we only have 2. And so our chi-square value is 48.4. Now we need to compare that to the critical value. So the critical value is 3.85, and we look for the intersection of that 1 degree of freedom and a probability of 0.05, and we get our critical value. So now we can decide what to do. We are going to reject our null hypothesis because this chi-square value is greater than the critical value. What that means is the lectin treatment has more cells in mitosis than we expected. What it means for the cell or the experiment is that lectin has an effect on cell division. It increases it. Okay, so go back and try the calculation on your own. You can stop it here and work out those details. All right, so let's get some practice with this. A student collected samples of cells from a woody plant during the spring and produced the drawings below. Sample 1 was collected from the woody stem of a plant, and sample 2 is collected from the root tip of the plant. Cells in different steps of mitosis are labeled with letters A, B, C, D, and E in sample 2. This is a 4-part question. So in part 1, you're asked to describe the role of mitosis in the growth of a plant. So why do plants divide? Via mitosis. Hopefully, a description would be one that included plant growth occurs when new cells are produced, or that mitosis is a process that produces 2 genetically identical daughter cells, which can develop into different tissues or divide further, creating that growth. Okay, part B, you're asked to explain why there are more cells observed in mitosis in the root tip than in the woody stem. And if you were paying attention to that diagram before, we know that only growth of root tips occurs in the tips, and that very little new mitosis or new cell development or new growth occurs in those woody stems once they are formed. Part C is asking you, and it's kind of a weird way of saying it, but it's asking you to refine the model in the space provided for your response by using the letters from 4 of the cell labels of sample 2 to represent the sequential steps of mitosis. All right, so you've got a model, and the model is telling you we go from prophase to metaphase to anaphase to telophase, and now you're asked to refine it by putting in pictures that represent those different stages. So think about prophase. In prophase, the chromosomes are condensing. So notice that you've got cell A, cell B, cell C, cell D, and cell E. Which one looks like prophase to you? So hopefully you recognize that it's E. Metaphase, remember that chromosomes line up in the middle of the cell. That looks like D. In anaphase, the chromosomes are pulled apart to opposite sides of the cell, and that looks like B, and finally telophase, where you start to see that in this case, in a plant cell, you'd start to see the beginning of a cell plate, which would develop into a cell wall, and that looks like cell C. So now we've refined that model. And the last part of this is to explain how the arrangement of cellular components during the step of mitosis portrayed by cell D in sample 2 facilitates the proper distribution of chromosomes in the 2 daughter cells. So let's go back and look at cell D. Cell D is metaphase. So they are asking you to describe what is going on in metaphase to make sure you get the correct distribution of chromosomes in those new daughter cells. So hopefully remember our conversation about the spindle microtubules. And what happens is that we need to make sure we discuss the fact that those chromosomes are lining up across the equator and that the spindle fibers enable the 2 chromatids of each chromosome to move to opposite ends of the cell before the cell divides. And that ensures the correct complement of chromosomes in each new cell. Let's do one more practice. This has to do with regulation. So we're looking at a diagram of the role of p53 in regulating the cell cycle progression in response to DNA damage. And this looks a lot like the example I gave you. So it says that DNA is damaged, that stimulates those protein kinases, and it looks like it's going to p53, and p53 leaves the nucleus. Well, p53 doesn't leave the nucleus, that it activates something on the DNA to produce that cyclin-dependent kinase, the CDK inhibitor. Okay, so you are asked first, this is a two-part question, that a skin cell completes one round of the cell cycle. Describe the products. Okay, so it doesn't really have to do with the figure yet. So one round, what's going to happen? The cell divides and you get 2 diploid cells that are identical, 2 genetically identical sets of DNA in each of those 2 diploid cells. In part B, you're asked to explain how p53 regulates the cell cycle in the presence of damaged DNA. So if there's damaged and you have p53, what's going to happen? Hopefully, you know that the cell cycle is stopped, and that's because p53 stimulates the expression of that CDK inhibitor, and that halts the cell cycle so that you don't continue to make cells that have damaged DNA. Oh, this actually has more parts. Sorry, I forgot about this part. So now you're asked to draw an X on the template in the space provided for your response to indicate the phase during which replication of the damaged DNA would occur. So which phase does DNA replication or duplication take place? S is correct. All right, and then finally, in part D, based on figure 1, explain how a mutation to the p53 may lead to an increased risk of cancer. So now you don't have p53. So what's going to happen? It can't make that CDK inhibitor, and the cell cycle will continue, and it will progress through the cycle with that damaged or mutated DNA, and that is going to continue without breaks. And basically, cancer is mitosis without breaks. All right, so a quick check for understanding. Again, this has to do with predicting the cause or effect of change or disruption to a component of a biological system. And this is based on the concept of cell signaling and regulation of the cell cycle. So you've got figure 1, MDM2 and P53, signaling in response to DNA damage controls cell cycle progression. The P53 and MDM2 proteins are involved in DNA damage response pathway that's shown above. Oh, and by the way, as you're looking at that, these mean a negative or inhibit. This is positive or stimulates. Okay? So in the absence of DNA damage, the MDM2 mediates the degradation of P53. In the presence of DNA damage, P53 is not broken down and forms active tetramers that stimulate the expression of genes that block cell cycle progression. Red blood cells are produced by the division and differentiation of multipotent stem cells. Researchers have observed that mice with a partial loss of function mutation in the MDM2 gene have a lower concentration of red blood cells in the blood than do mice that express the wild-type MDM2 allele. Two task verbs here. Predict how a loss of function mutation in the P53 gene is most likely to affect the cell cycle in response to DNA damage. To make that prediction, and then justify your predictions. So again, we don't have P53. What's going to happen to the cell cycle? Make a prediction. And then justify that. Without P53, what happens? Okay, so the cell cycle will proceed. We just said that without P53, we can't make the inhibitor, cell cycle goes. And the justification is that it cannot regulate the cell cycle. You could be that blanket statement, or you could be more detailed. All right, so your task verb in that last part was justify. So remember that justification means give a good reason for something or show it to be right. All right, so here's your debrief. You should now be able to describe the events that occur in the cell cycle. You should be able to explain how mitosis results in the transmission of chromosomes from one generation to the next. You should be able to describe the role of checkpoints in regulating the cell cycle and describe the effects of disruptions to the cell cycle on a cell or organism. You should also be able to do a chi-square analysis and to do science skill B, which is to predict the cause or effect of a disruption to a event that's happening within the cell or organism. Your key takeaway for this topic is that heritable information provides the continuity of life. All right, let's take a look at some common errors and misunderstandings, and then I'm going to give you your homework. So first of all, I'm going to give you an FRQ, and this was given years ago at on an AP exam, so you may have seen this by looking at practice questions. Here's the question. It asked you to, it gave you a statement that the cell cycle is fundamental to the reproduction of eukaryotic cells, and then it asked you to describe how the cell cycle is regulated and then discuss one consequence of abnormal regulation. Okay, so the scoring guidelines are divided into regulation and consequence. So for regulation, there needed to be a description of the checkpoints and make sure that all three of those checkpoints were discussed. In your description, the action of MPF and Cdks needs to be discussed in relation to checkpoint regulation. You also needed to talk about contact inhibition. Those are external factors, that density independent inhibition. And then hormones or growth factors. I gave you the example of the platelet-derived growth factor. So all of those things needed to be discussed. In terms of the consequence, there would be a couple of different things to key in on. One, of course, is cancer, that there would be, if the cell cycle is not regulated, that the mitosis cannot stop and the cells continually divide, as seen in cancer. Apoptosis, which is programmed cell death, is another result of abnormal cell cycle control. And then the third thing to talk about would be anything that happens as a result of chromosomes not getting to the right place, right? So you can have too many chromosomes or too few chromosomes. So you have polyploidy or aneuploidy. Or non-disjunction, where the chromosomes fail to separate. And we'll talk more about that tomorrow when we talk about meiosis. All right. So here's what happened. Here's the common errors. Students had difficulty explaining the action of MPF and what CDKs do. So go back and look at both of those things. Students often omitted discussion of contact inhibition. So you need to talk about those external factors as well as internal factors. And also students failed to discuss growth factors in the regulation of cell cycle. So those last two are external factors. The first one are the internal checkpoint system. Okay. All right, so some practice. This is homework tonight. It's pretty easy breezy because I don't want to spend a whole lot of time with the warm up tomorrow, because tomorrow we're going to be doing meiosis and we have a lot to talk about there. So I'm just asking you to identify the 3 stages, major stages of interphase, describe the sequence in which they occur, and you should be thinking about what happens in each of those. And if you wanted to get fancy, you could even talk about where those checkpoints occur in interphase. Okay, so that's homework due tomorrow. I have a couple of reminders for you. So I have an exam prep checklist for you. The first thing is device and connectivity. So you need to have a device to take your exam on. The exam again is on Monday, May 18th at 2 p.m. Eastern Standard Time. So you need to make sure you have a device by that time. You also need to make sure you have reliable internet. Both of these things you can be helped by the College Board. If you need help with either a device or connectivity, please reach out directly at cb.org/tech, and you need to do that by this Friday, April 24th. Also, make sure that the College Board has your correct email address, and you can do this by checking at your profile on AP Central. And then finally, let your AP Bio teacher in on your plans, whether or not you plan to take the test or not take the test, just so that gives them an idea of how many students are actually going to be sitting for the exam, and also if you need any, additional review for the exam. Okay, so thanks for watching today. I hope you'll join us again. Be sure to complete that practice question. It's an easy one tonight, and we'll review the answer in tomorrow's lesson, which will be on meiosis. So I'll see you guys tomorrow. Thanks for watching.